Cells use energy stored in food, such as glucose, to make ATP. Every single organism requires a steady food supply in order to survive. A bird eats a caterpillar, the caterpillar ate the tree's leaves, and the tree made its own food by that photosynthetic process. All plants and animals, as well as many microbes, which are those single-celled microorganisms, use foods such as glucose and oxygen gas to produce ATP. ATP is that energy carrier that's used to power most cellular activities. The process of using glucose and oxygen to produce this ATP is called aerobic respiration. And today, we're going to spend most of our time figuring out how aerobic respiration works. The overall equation is as follows. We're going to take a sugar and we're going to extract that energy to make ATP. We can see we've got glucose plus oxygen as the inputs and carbon dioxide, water, and energy as the outputs. In breaking down our sugar, we're going to be able to have energy transformations. We're going to be able to move around those components to ultimately release energy that will be in the form of ATP. Also notice that oxygen is part of this input because oxygen loves electrons. Oxygen pulls those electrons towards it. Remember from our water lecture, we know that water molecules form hydrogen bonds between all of those water molecules because oxygen loves to pull electrons towards it. Cellular respiration is directly linked to breathing in organisms such as humans. Inhaled oxygen is consumed during cellular respiration, and carbon dioxide, which is simply produced as a byproduct, is exhaled into the atmosphere. We breathe in oxygen to be used during cellular respiration, and we breathe out carbon dioxide, which is the waste product. Cells use the ATP that's formed during respiration to do work, such as muscle contraction. That's why when you're running or working out, you have to breathe more, right? You need to get more oxygen into your lungs so that you can continue to make that ATP. So let's take a look at this question. Do plants carry out cellular respiration? You've got several choices. No, photosynthesis has the same function in plants as respiration has in animals and microbes. The next choice is no, their energy needs are too small to require respiration. The next choice is yes, they require ATP like other living things, and respiration generates ATP. And your final choice is yes, they require cellular respiration as a way to get rid of their extra carbon dioxide. So pause the lecture now if you don't want to see the answer. The only correct answer is yes, plants require ATP just like other living things, and cellular respiration is that process that generates this ATP. When we're studying for the exam, make certain that you understand how cellular respiration is related to breathing. Many of the reactions of cellular respiration occur in the mitochondria. And recall that a mitochondrion is one of those organelles that we find in eukaryotic cells. Mitochondria have two phospholipid bilayers, an outer membrane and an inner membrane. Many enzymes are found along the inner membrane, thus catalyzing the reactions of our electron transport chain. Between the mitochondrial membranes is what we refer to as the intermembrane compartment. The space within the intermembrane compartment is this mitochondrial matrix. And this matrix is essential because it is the place where the reactions of the citric acid cycle will occur. Another name for the citric acid cycle is the Krebs cycle. All right, let's make sure we understand the anatomy of a mitochondria. Where is the mitochondrial matrix? Is it outside the outer membrane, between the inner and outer membranes, or inside the inner membrane? Pause the lecture if you don't want to see the answer, and the answer is inside the inner membrane. When you're studying for the exam, make certain that you can describe which respiratory reactions occur in each part of the mitochondrion. So before we go any further, let's review how electron carrier molecules work. Remember that electron carriers are those molecules that can capture the electrons and the energy contained within those electrons. During the light reactions of photosynthesis, we learned about NADPH and how those electrons are loaded into our NADP plus to give us NADPH. Well, that's just one of the electron carrier molecules. You can see the names of several others here, NADH and FADH2. There's several other types of electron carriers, but at the end of the day, they are all essential for picking up electrons and then carrying those electrons to where they are needed. Electrons can be deposited, energy and all, with another molecule to drive another reaction. And typically the reaction that is driven is the synthesis of ATP. To help us keep track of all of the stages of cellular respiration, we have a roadmap. And this roadmap is for the path of a single glucose molecule during the cellular respiration process. And notice we start outside of the mitochondrion. The first part of cellular respiration is called glycolysis, and glycolysis occurs outside in the cytosol, that fluid matrix that surrounds all organelles. Glycolysis occurs in this fluid matrix, and what does it do? It takes our glucose. Remember, glucose was produced during the carbon reactions of photosynthesis, and it's going to break down that glucose into 2 pyruvic acids. In addition, glycolysis is going to make a little bit of ATP and a little bit of NADH, which is one of these electron carrier molecules, fully loaded with electrons. During glycolysis, a glucose molecule is split into 2 3-carbon pyruvates. And this is kind of easy to visualize because remember, glycolysis has 6 carbons. So we can divide 6 into 2 3-carbon molecules. And we refer to these 3-carbon molecules as pyruvates. Enzymes, remember, without enzymes, cells don't get anything done. The enzymes of glycolysis extract some of the potential energy that's stored in our glucose sugar. This process yields a little bit of ATP and a little bit of electron-carrying NADH molecules. So not a lot, but enough to get a little bit of work done. Glycolysis requires an input of 2 ATPs in order to initially activate our glucose. So you might have heard of the phrase, you've got to spend money to make money, right? So the cell's got to spend a little bit of ATP in order to make a little bit of ATP. In total, 4 ATP are produced after one round of glycolysis, but 2 were used to start the reaction, so therefore the overall net yield of glycolysis is simply 2 ATPs. Note that these reactions of glycolysis do not require oxygen. Remember that oxygen is required for aerobic cellular respiration, but not in the very first stage, because glycolysis is the first stage and no oxygen has been used. Glycolysis can therefore occur in conditions that lack oxygen. And the fancy word for conditions that lack oxygen is anaerobic. And you can see this word on the screen in front of you. In summary, glycolysis splits our glucose, thus rearranging those carbons into 2 pyruvates. In addition, it yields a little bit of ATP and a little bit of electron carrier molecules. All right, let's review. If 8 glucose molecules enter one round of glycolysis, the net products will be how many pyruvates and how many ATPs? Your first choice is 2 and 2. Your next choice is 4 and 4. Your next choice is 8 and 8. And your final choice is 16 and 16. Pause the lecture if you don't want to see the answer. The correct answer is if 8 molecules of glucose enter glycolysis, the net products will be 16 pyruvates and 16 ATPs. All right, so now we've covered glycolysis. Now we move into our mitochondrion for the second step of cellular respiration, which is called the citric acid cycle. The citric acid cycle is also referred to as the Krebs cycle, so we can use these names synonymously. It's named after the scientist that discovered it. Where does it occur? It occurs in the matrix of our mitochondria. And what does it do? It needs water, and it needs those pyruvic acids, those pyruvates that we made during glycolysis. And it's going to give off the waste product of carbon dioxide. It produces a little bit of ATP and quite a few of these electron carrier molecules. That is to say, it loads quite a bit of electrons into the electron carriers because we're going to need a lot of electrons in the next stage of cellular respiration. The reactions of the citric acid cycle and the next stage, the electron transport chain, require oxygen. These reactions give us much more ATP than glycolysis alone. The 2 pyruvate molecules that were produced during glycolysis undergo an oxidation reaction as they enter the mitochondrion. Recall that oxidation equals a loss of electrons. Oxidation reactions are going to therefore release energy. A carbon atom is stripped from each of our pyruvates, and then these leave the cell as carbon dioxide gas. Remember, carbon dioxide gas is the waste product of this second stage of aerobic respiration. At the same time, NAD+ is reduced to NADH. Remember that when we reduce something, we give it electrons. So we're loading electrons into our electron carrier molecule. Through this process, each pyruvate, now with only 2 carbons, is converted into a special molecule called acetyl CoA. Now the CoA part stands for coenzyme A. And this is going to prep the pyruvate to get it ready for the citric acid cycle proper. So far we've just been talking about getting ourselves ready for the citric acid cycle. And to get ourselves ready, we need to take our pyruvates and convert them into acetyl CoA. Acetyl CoA has only 2 carbons because 1 carbon from the original pyruvate left the organelle as carbon dioxide. That carbon dioxide gas, which is the waste product during this stage. Each acetyl CoA molecule can then enter into the citric acid cycle proper. During the citric acid cycle, these 2 acetyl CoA molecules are oxidized, which means they lose electrons, and that is going to allow us to yield a little bit of carbon dioxide, which is a waste product, a little bit of ATP, and we're going to be able to use this energy to load tons of electrons into our electron carrier molecules. So far, aerobic respiration of just 1 single glucose molecule has yielded only 4 little ATP, so that's not very much. But we've made tons of electron carrier molecules. We've loaded tons of electrons into our NADH, for example. We can use these electron carriers in the next stage of our respiration reactions. The final stage is the electron transport chain, and the electron transport chain gives us the most bang for our buck. We're going to produce a ton of ATP at the end of electron transport. Where does this occur? It occurs on the inner membrane. Specifically, we call this the cristae of the mitochondrion. What happens? Well, we have an electron transport chain, which we know is just a series of proteins that are embedded in the membrane. And we're going to have the electron carriers fully loaded. We're going to take these electrons, we're going to put them in the chain, and we're going to use this organized movement of electrons down the chain in order to pump protons into the intermembrane space, thus creating a proton concentration gradient. We know that things move from high to low concentration. Those protons move through a special machine, thus building tons of ATP. Oxygen is used during this part of the reaction, and water is produced. Electron transport releases the energy that our cells need to make the most ATP. Looking in our textbook, we can see that our electron carrier molecules are going to donate their electrons to the beginning of an electron transport chain. As electrons travel in an organized manner through the transport chain, carrier molecules will use the potential energy of the electrons to transport hydrogen ions, which we refer to as protons, into the intermembrane compartment. These protons will then build up in high concentration, and we can use those later on to build ATP. At the end of our transport chain, electrons are donated to oxygen. Oxygen pulls those electrons towards it. In a sense, we can think of it as an anchor, pulling electrons down the chain. At the end of that chain, those electrons combine with hydrogens to ultimately form water. So we can use those electrons to build water, H2O. And that water is again a waste product that can be used by another metabolic reaction. Hydrogen ions move down their concentration gradient from the intermembrane compartment into the matrix through a special little machine called the ATP synthase, thus producing ATP. Please note that this is just like the light reactions. The same process occurred on the electron transport chain that happened in the first stage of photosynthesis. At the end of this process, the electron transport chain produced 34 ATP, so that's a lot more ATP than we got in the previous steps of cellular respiration. When you're studying for the exam, make sure that you understand how these electron carrier molecules ultimately power this ATP formation. Overall, biologists can say that cellular respiration of one glucose yields 36 ATPs. Glycolysis and citric acid cycles each produce 2, and electron transport chains produce 34. But remember that we had to spend money to make money. We had to input 2 ATPs at the beginning, so our overall net yield is about 36. Now, it's easy to imagine one single mitochondrion making 36 ATP every second. But consider that human muscle cells have up to 10,000 mitochondrions per cell. And there may be tens of millions of cells in one muscle tissue. That is a huge amount of ATP being produced every second in a working muscle. Here's a question for you. What happens to glucose's carbon atoms during the overall process of aerobic respiration? The first choice is they are donated to oxygen. The next choice is they remain in the pyruvate molecules. The next choice is they become part of ATP. And the final choice is they are released as carbon dioxide. Pause the lecture if you don't want to see the answer yet. The only correct answer is they are released as carbon dioxide. Now we can't say B because pyruvate is prepped into an acetyl CoA form. Therefore, we can only say D, they are released as carbon dioxide. When you're studying for the exam, make certain that you can describe all 3 stages of cellular respiration. Proteins and fats are also used as energy sources for the cell, but please note that these molecules enter our energy extracting pathways in slightly different places, but overall they can still help produce ATP for our cell. Now, all of this has been about oxygen, right? We need oxygen to pull those electrons down the electron transport chain. But organisms can produce ATP in the absence of oxygen as well. We refer to this as fermentation. Recall that glycolysis produces ATP and it does not require oxygen. However, glycolysis does require NAD+, because we need to load electrons into this electron carrier molecule. And NAD+ is recreated during the electron transport chain of cells that are undergoing respiration. In the absence of oxygen, a cell can recreate this NAD+ by another pathway, and this is what we refer to as the fermentation reaction. In alcoholic fermentation, NADH reduces pyruvate into ethanol. So ethanol is that waste product, and NAD+ is therefore recreated. In lactic acid fermentation, which occurs in our muscles, NADH reduces the pyruvate to lactic acid. That lactic acid builds up in your muscles and can cause you to feel sore. NAD+, however, is recreated, so it allows us to make just a little bit of ATP. During fermentation, oxidation of a glucose molecule yields a very small amount of ATP. It's not much, but it can sustain work for very short periods of time in the absence of oxygen. Here's another challenge question. What is the main advantage of fermentation over aerobic cellular respiration? The first choice is fermentation generates ATP even if oxygen is not present. Fermentation generates more ATP per glucose than aerobic cellular respiration. Fermentation does not generate toxic byproducts such as carbon dioxide, or fermentation gets rid of pyruvate, which would otherwise accumulate in the cell. Which one is the main advantage? Please pause the lecture if you don't want to hear the answer. Fermentation generates ATP even if oxygen is not present is the only advantage here of fermentation over aerobic cellular respiration. So our textbook ends with kind of a fun case study, and I couldn't resist showing you this one since it has such a provocative title, Plants' Alternative Lifestyles Yield Hot Sex. So that's kind of funny. But it talks about this plant called philodendron, which is able to warm its flowers. How does it warm these flowers? Well, the electron transport chain prematurely transfers electrons to oxygen. Instead of extracting the remaining potential energy in those electrons, the energy is lost as heat, thus heating up that flower. In this case, this inefficient electron transport chain actually benefits the plant because these beetle pollinators are attracted to the heat. If the flower did not heat up, the beetles wouldn't come and it wouldn't get pollinated. And if plants don't get pollinated, then they're not able to make seeds in most cases. Beetles also benefit from this heat. Warm beetles use less energy, and the energy they save can be used for finding food or mates, meaning they can pollinate more flowers. The hot flowers, therefore, increase the reproductive success of both beetles and the plant.